base, and is called the intralobular vein. At the base of the lobule, this vein opens directly into the sublobular vein with which the lobule is connected. The sublobular veins unite to form larger and larger trunks, and end at last in the hepatic veins. These converge to form three large trunks, which open into the inferior vena cava, while that vessel is situated in its fossa on the posterior surface of the liver. 3. The bile ducts commence by little passages in the liver cells, which communicate with canaliculi, termed intercellular biliary passages, bile capillaries. These passages are merely little channels or spaces left between the contiguous surfaces of two cells, or in the angle where three or more liver cells meet, and they are always separated from the blood capillaries by at least half the width of a liver cell. The channels thus formed radiate to the circumference of the lobule and open into the interlobular bile ducts, which run in Glissen's capsule, accompanying the portal vein and hepatic artery. These join with other ducts to form two main trunks, which leave the liver at the transverse fissure and by their union form the hepatic duct. Structure of the ducts. Walls of the biliary ducts consist of a connective tissue coat, in which are muscle cells arranged both circularly and longitudinally, and an epithelial layer, consisting of short columnar cells resting on a distinct basement membrane. Excretory apparatus of the liver. The excretory apparatus of the liver consists of 1. The hepatic duct, formed by the junction of the two main ducts, which pass out of the liver at the porta; 2. The gallbladder, which serves as a reservoir for the bile; 3. The cystic duct, or the duct of the gallbladder; and 4. The common bile duct, formed by the junction of the hepatic and cystic ducts. The hepatic duct, ductus hepaticus. Two main trunks of nearly equal size issue from the liver at the porta, one from the right, the other from the left lobe. These unite to form the hepatic duct, which passes downward and to the right for about 4 centimeters between the layers of the lesser omentum, where it is joined at an acute angle by the cystic duct, and so forms the common bile duct. The hepatic duct is accompanied by the hepatic artery and the portal vein. The gallbladder, vesica phellia. The gallbladder is a conical or pear-shaped muscular membranous sac lodged in a fossa on the under surface of the right lobe of the liver, and extending from near the right extremity of the porta to the anterior border of the organ. It is from 7 to 10 centimeters in length, 2.5 centimeters in breadth at its widest part, and holds from 30 to 35 cubic centimeters. It is divided into a fundus, body, and neck. The fundus, or broad extremity, is directed downward, forward, and to the right, and projects beyond the anterior border of the liver. The body and neck are directed upward and backward to the left. The upper surface of the gallbladder is attached to the liver by connective tissue and vessels. The under surface is covered by peritoneum, which is reflected onto it from the surface of the liver. Occasionally, the whole of the organ is invested by the serous membrane, and is then connected to the liver by a kind of mesentery. Relations. The body is in relation by its upper surface with the liver, by its under surface with the commencement of the transverse colon, and farther back usually with the upper end of the descending portion of the duodenum, but sometimes with the superior portion of the duodenum or pyloric end of the stomach. The fundus is completely invested by peritoneum. It is in relation in front with the abdominal parietes, immediately below the 9th costal cartilage, behind with the transverse colon. The neck is narrow and curves upon itself like the letter S. At its point of connection with the cystic duct, it represents a well-marked constriction. Structure. The gallbladder consists of 3 coats, serous, fibromuscular, and mucus. The external or serous coat, tunica serosa vesicae phelii, is derived from the peritoneum. It completely invests the fundus, but covers the body and neck only on their under surfaces. The fibromuscular coat, tunica muscularis vesicae phelii, a thin but strong layer forming the framework of the sac, consists of dense fibrous tissue, which interlaces in all directions, and is mixed with plain muscular fibers, disposed chiefly in a longitudinal direction, a few running transversely. The internal or mucus coat, tunica mucosa vesicae phelii, is loosely connected with the fibrous layer. It is generally of a yellowish-brown color, and is elevated into minute ruggi. Opposite the neck of the gallbladder, the mucous membrane projects inward in the form of oblique ridges or folds, forming a sort of spiral valve. The mucous membrane is continuous through the hepatic duct with the mucous membrane lining the ducts of the liver, and through the common bile duct with the mucous membrane of the duodenum. It is covered with columnar epithelium and secretes mucin. In some animals it secretes a nucleoprotein instead of mucin. The cystic duct, ductus cysticus. The cystic duct, about 4 cm long, runs backward, downward, and to the left from the neck of the gallbladder, and joins the hepatic duct to form the common bile duct. The mucous membrane lining its interior is thrown into a series of crescentic folds, from 5 to 12 in number, similar to those found in the neck of the gallbladder. They project into the duct in regular succession, and are directed obliquely around the tube, presenting much the appearance of a continuous spiral valve. When the duct is distended, the spaces between the folds are dilated, so as to give to its exterior a twisted appearance. The common bile duct, ductus choledochus. The common bile duct is formed by the junction of the cystic and hepatic ducts. It is about 7.5 cm long and of the diameter of a goose quill. It descends along the right border of the lesser omentum, behind the superior portion of the duodenum, in front of the portal vein, and to the right of the hepatic artery. It then runs in a groove near the right border of the posterior surface of the head of the pancreas. Here it is situated in front of the inferior vena cava, and is occasionally completely embedded in the pancreatic substance. At its termination it lies for a short distance along the right side of the terminal part of the pancreatic duct, and passes with it obliquely between the mucous and muscular coats. The two ducts unite and open by a common orifice upon the summit of the duodenal papilla, situated at the medial side of the descending portion of the duodenum, a little below its middle, and about 7 to 10 cm from the pylorus. The short tube formed by the union of the two ducts is dilated into an ampulla, the ampulla of Vater. Structure. The coats of the large biliary ducts are an external or fibrous, and an internal or mucous. The fibrous coat is composed of strong fibroareolar tissue, with a certain amount of muscular tissue, arranged for the most part in a circular manner around the duct. The mucous coat is continuous with the lining membrane of the hepatic ducts and the gallbladder, and also with that of the duodenum, and, like the mucous membrane of these structures, its epithelium is of columnar variety. It is provided with numerous mucous glands, which are lobulated and open by minute orifices scattered irregularly in the larger ducts. End of section 24.